Thanks so much for having me. I was told that this talk was supposed to stretch people's brains, so I think by the end of this 45 minutes, it will be stretched like a noodle because I'm really not a fit here. I'm a chemist in biology department, so I'm used to being a misfit, but now I'm a chemist among computational biologists, so we'll see how that goes. But what I want to talk about today is using biology to do things that we normally usually associate with computational systems. And why do we want to do it? How are we doing it? And what's the point and where this is going? So my pointer disappeared. Let me do something to get it back. We had a pointer when we did a test right before this, but that might be, oh, yes, all right, awesome. There's always this question, how many PhDs does it take to put slides up on the screen? So, okay, the first step is done. Okay, so what is that we're trying to do is, okay, so this works, but well, apparently more than one because I can't change my slide. I can't move my slides at all. Let's see. I'm not doing a chalk talk. Okay, that works. So let's see if it's going to keep working. Yes, it keeps working now. See, the organizer had to come up and scare the computer. Okay, so this is kind of what we want to do is we want cells to pretend to behave like Boolean logic gates. It's not a new idea. People have thought about doing this for quite a while. The idea for doing it is biology can regenerate, biology is sustainable, whatever that means. And also biology interacts with biology, goo mates with goo. And if you think about using computational systems to do things like body-computer interfaces, brain implants, any kind of neural system implants, any kind of muscle implants, all of that is really difficult, technologically difficult right now because tissue is scarce when it mates with electronics, mates with silicon devices. The idea is that if we can make biological systems to process information in a binary way like an electronic system, then we can conceivably develop something like an implant that will mate with a body. There's also a lot of applications in space exploration, in autonomous applications. The idea is that if you have something that heals, that can self-organize, then you can send it to Mars or for Artemis missions right now, or for any other kind of a remote exploration applications. You can have a device that will work for much longer, and if it gets hit with something with a physical or radiation threat, it can repair itself, it can reorganize itself. This obviously sounds like a science fiction and we're not there yet. I'll show you a little bit of where the science is right now and what's the technology that we're using to get there. But basically that's the idea. If biological molecules can behave like circuits, like logic gates, then we can do a lot of cool stuff. The problem is that unfortunately biological systems, cells don't usually want to cooperate with us. And they have this huge evolutionary baggage that compels them to first replicate, evolve, make more copies of themselves to stay healthy. And then if they have any resources left, they can run my logic gate circuit. And that's been a big problem with biocomputing ever since that idea even came up, is that you can build a genetic circuit that will behave like a Boolean logic gate. You can put it in bacteria and bacteria will chew up on it, and in a few generations you will have a very leaky circuit. And that's been a problem that we as biological engineers have been trying to solve for quite a while, still rather unsuccessfully. So here's the idea that for the simplest biological circuit, you have a piece of DNA. We always use DNA because that's easy to program, easy to synthesize. Out of that DNA comes a protein, and that protein is usually fluorescent. That's the idea behind the simplest biological genetic circuit that has been first tried and has been now tried to death in many different organisms. That's kind of what the principle is. You start with DNA and your output is some sort of a fluorescent or other signal that came out of the protein. And so this is your simple circuit. Now you can scale it up. The problem, though, is that if you have that circuit and if you, one sec, let me put that notification away. Okay. If you have that circuit and if you then drop it into your target organism, that is a cell, there are problems. Problems with cell not being able to reliably replicate that circuit and also reliably perform operations that that circuit requires, because cells have metabolism. And the problem with cell metabolism is that we don't actually understand it. Despite all of the years that biologists have been poking at cells, it's still a black box. We still don't have a full chemical component list of any living cell. We know a genome of a minimal cell, the Craig Venter's mycoplasma-based synthetic cell, but that genome still, despite us knowing the whole sequence of that genome, still contains a couple dozen of genes, those so-called essential genes of unknown function. So essentially, it is a black box. So we're putting this very complex, fine-tuned genetic circuit into what is something we don't really understand, a black box. So the business model of my field of engineering synthetic cells is that let's make the chassis simpler. Let's stick to the design of the biological circuit. Let's say, okay, we still have DNA as a base and our readout is fluorescence, but let's make the surrounding environment much simpler and more controllable. And that's what the synthetic cell is. It's a cell-like entity that has some but not all functions of a living cell, and it allows us to do those computational operations with much higher fidelity and accuracy because the synthetic cell is dumb, dead as a doornail. It does not evolve. It does not go on and does its own thing. It doesn't have epigenetic changes that accumulate with every generation. So we can basically, at least hope, that's the theory, we can perform those operations over a long period of time with much higher fidelity. So there are two different ways in which we go about making synthetic cells. So we have those in vitro systems, molecules, purified, well understood molecules. On the other hand, we have those living cells. If you put your circuit into a living cell, it will be leaky. If you put it into an in vitro system, it will be very programmable, very high fidelity. But the problem is that in most of the genetic circuits that are done in vitro, so in like when you hear about people talking about in vitro genetic circuits, that's usually a single protein being translated. So one molecule is basically one photon of light. So if you want to have a strong signal, you don't have any way of amplify your signal. Living cells will amplify your signal, but you'll pay for it in that leakiness. Now, if you want to do more, if you want to scale that up, living cells will oblige. In vitro systems will not replicate, will not amplify. Also, if you want to change your program, if you want to make any modifications, living cells evolve, so your circuits get leaky change over time. In vitro circuits stay the way you designed them. Also, in vitro, you can use whatever you want, whatever chemistry you want. Those of you in the audience, if there's any chemist in the audience, you're aware of the fact that nature is really very selective in terms of the molecules that it uses. It's really, from the chemical point of view, really boring. Nature only uses 22 proteinogenic amino acids, only 5 nucleobases. That's only a tiny fraction of what we can actually throw at those circuits to really do combinatorial programming using chemistry. In vitro, you can use whatever chemistries you want. You have that flexibility. And then in vitro systems, you can lyophilize, put them on the shelf and forget about them. Living cells are not very happy to do that. If you want to store your circuits for a longer period of time, you have to freeze it, assuming it's an organism that survives freezing. So this is where synthetic cells come in. We kind of combine the best of both worlds. We combine the benefits of living cell engineering. So we have signal amplification, we have the ease of programming. We also have the ability to control the system through kind of a multi-layered cascade of signals. The one thing we don't have right now is self-replication, so you still have to build the system every time. It's a bug when you think about it as a biologist, but you can also think about it as a feature, because if they don't replicate, then they don't evolve. Obviously, ideally, you would have something that replicates without evolution, and that's something that our field is working towards. So genetic circuits can be made in a synthetic cell, and I'll show you a couple examples in a few minutes. But mind you, I still haven't told you what really is a synthetic cell. And the reason I haven't told you that is because nobody knows. We don't have a good definition of what a synthetic cell is, because everyone in the field applies their own definition to what they're doing. Obviously, I believe that what I make in my lab is the one only true synthetic cell. My colleagues disagree. So the simplest definition that I can give of a synthetic cell is something that has not been alive before the experiment started. So when you think about it, all life on Earth that you see right now comes from an unbroken line of life. There has never been a second origin of life since the first life started on Earth 3.8 billion years ago. And what we want to do is we want to break that cycle. We want to take something that's not alive when I start my experiment and make it into a lifelike system that is the synthetic cell. So we're basically breaking that constant line of dividing organisms, constant line of life. People love definitions. They always push me for definitions. So here's my definition. A synthetic cell would be something that's more than a sum of its parts. It has emergent properties. So it's not just simple chemistry. It's a lifelike system that has some emergent properties, but is still controllable. That's the biggest thing, is we have to be able to control our experiments, because otherwise we might as well just work with bacteria. So we have to control it, but it has to have some sort of a property that's not obvious from just looking at the ingredient list. And some of the things that allow us to engineer those properties are things that you cannot normally control in living cells. There is a lot of things in synthetic cells that we can control that are not possible to control in living cells. Things like the size of a cell, the identity of the chemistries that we're using. We can expand the genetic code. We can move away from DNA. We can use other types of nucleic acids. So you can think of a synthetic cell as sort of a biological reactor, kind of a bioreactor entity that's made out of biological parts, but not exactly living, not coming out straight from a line of life. So kind of like a cell made out of random chemistry parts. People always use Lego block analogs that we're basically putting molecules together into a lifelike system. So that's a general definition that I think most people in the field would, even if they have a little bit of a problem with it, they would agree that, okay, that sort of works. Now, this is a very specific definition for what I consider a synthetic cell in my lab. It's a liposome that contains central dogma, modified central dogma, and expresses proteins. I'm trained as a liposome biophysicist, so I really like to have a compartment. And when you think about biocomputing applications, you want to be able to separate your circuits, to segregate your reactions. So think of it as tiny little chips, tiny little units of that can perform different spatially separated computations. And that's what the membrane gives you. It basically separates your reactions. So the membrane of my synthetic cells is usually just phospholipids. It's the same phospholipids that make up your own membranes. They make nice, well-behaved bilayer membranes. Sometimes we use fatty acids. We add cholesterol. We love cholesterol. Membrane biophysicists love cholesterol because it makes our membranes fluid and really well-behaved. Sometimes we don't use liposomes. I'm required to put that image on this slide because my postdoc would be really angry if I don't. He's a microfluidics guy. He loves this. Microfluidics is great. We often use droplet microfluidics to prototype things, so it doesn't always have to be a vesicle. Here I said it. Moving on. What's really fun is what's inside the synthetic cell. That's what we really play with. That's what we use to program the synthetic cells. And that's a cell-free protein expression system. So proteins can be expressed outside of the cells. That's not news. But we also can express proteins that don't contain just the natural amino acids. We can expand that chemical repertoire of proteins. We can also expand the types of lipids that we're using. So we can combine all of those lifelike but not living properties into a cell system, cell-like system that then we can fully engineer. We still understand it because we put it together. Even though most of the components are purified from natural sources, we purify it so we understand exactly where every molecule goes. Okay, so I just told you that I'm putting together non-living molecules into a cell-like system. And some of you, those of you who have enough common sense, are probably thinking that I'm crazy right now, that this is never going to happen. There's just no way that this can work, right? So I want to show you a quote from an article that's been published in the New York Times over 100 years ago. Very respected publication. So the year is 1903. We as the society have been talking about the feasibility of building a flying machine heavier than air. So the year is 1903. We have dirigibles, we have balloons. They all fly because they're lighter than air. And so the common engineering paradigm is that in order to fly, you have to be lighter than air. You have to heat up the air or use some noble gas that's lighter than air. And so someone published an article in this very respected journal that says, don't bother even considering building a flying machine heavier than air. This will never work. And then 2 months later, the Wright brothers flew their first airplane. And I love this contrast because it shows you that if you tell scientists and engineers that something is impossible, we're just going to go out and do it. And that's where the field of synthetic cell engineering is right now, is A lot of people told us that it's impossible to build a lifelike system from non-living components. This cannot be done in the lab. This will never work. And so we're out there doing it. And now I'll show you a few examples of the state of the art where the field really is right now. So let me start with one project out of my lab that we recently wrapped up that shows example of biocomputing, of programmable biocomputing platform that's made out of a synthetic cell system. So we called it Trumpet. The reason we called it Trumpet is because the first widely used biocomputing platform that performed computations using genetic circuits was a system out of Chris Voigt's lab that they called Cello. And so ever since then, the biocomputing platforms are supposed to be named after instruments. So ours is Trumpet because the lead grad student on this project plays trumpet. So here's the trumpet. Obviously, once we came up with the name, we had to develop a backronym. So it is related to RNA transcription and whatnot. It's a trumpet. So what is a trumpet? A trumpet relies on using a genetic template, a DNA template, and then produce light. So we can perform Boolean logic gate operations, and the output is light. So how do we do it? So the normal way of doing genetic logic gates, the standard way that most people in the field use is you have a DNA template, then you transcribe that template into mRNA, and then you express a protein out of that mRNA. Designing Trumpet, we kind of stopped in the middle. We skipped translation, we stop at transcription. So our readout is a fluorescent piece of RNA, and the operations of the logic gate happen on the DNA template. So the template, the single-stranded DNA template is our logic gate template. That's where all the actual logic happens. And then to read out the state of your gate, you transcribe it, and you get light or you don't get light. So here's an example of one Trumpet gate that shows you how this works. So this is an AND gate. So we have a single-stranded DNA template that has a RNA polymerase promoter. This is a promoter for a T7 RNA polymerase. Those of you who got the mRNA vaccine for COVID are very familiar with T7 promoter. That's the promoter that's been used industrially to make most of the RNA that we use, including mRNA vaccines. T7 is a great RNA polymerase. I love T7. So there's a T7 promoter here. Then there's the logic gate area. So this is the area that actual logic gate operation will happen. And then there's the readout. The fluorescent aptamer is how we actually read out the signal from the gate. So RNA can be fluorescent, not only proteins are fluorescent. RNA can be fluorescent, and those fluorescent pieces of RNA are called aptamers. They're named after vegetables. The one we're using here is broccoli. The first one developed was spinach. There's also corn. Some of the things we use in this work is corn and pepper. There's also a mango, which really bugs me because that's not a vegetable. Fluorescent proteins are named after fruits. So when they started making fluorescent RNA, they started naming them after vegetables. But mango, come on. Okay, so here's a logic gate, single-stranded DNA logic gate. And if we have two inputs, our inputs into the logic gate, so the signals that come in are short oligos, short single-stranded oligos, think primers. They're about the length of a typical PCR primer. So two of those primers come in, and so our template here is single-stranded. Our two input oligos come in, and now our template is double-stranded. So that region here with two input oligos now becomes double-stranded. And what happens when a piece of DNA becomes double-stranded is that now it can become a target for a restriction enzyme. Restriction enzymes are enzymes that chew up DNA. And they chew it up in a very sequence-specific way, and many of them are extremely particular about chewing up double-stranded DNA only. You feed them single-stranded DNA, nothing happens all day. You feed them double-stranded DNA, it gets cut up immediately, the same sequence. So one of those enzymes is what we use here. We're using a protein restriction enzyme that's very specific to double-stranded DNA. So now if our logic gate region became double-stranded because we provided two inputs, now the restriction enzyme can bind and cut. And if it cuts, it separates the template region that has the promoter from the template region that has the aptamer. And without the promoter region, RNA polymerase cannot transcribe. So if you cut, you have no signal. So if that cut happens, the promoter is separated from the aptamer region and there is no signal if you transcribe it. Now, if you have one input or no input, the region becomes fully or partially single-stranded. And if it's fully or partially single-stranded, the restriction enzyme cannot digest it, which means our template remains intact. And that means our transcription reaction can produce fluorescent signal. So 2 inputs, the signal is 0. 1 or no inputs, the signal is 1. That's a truth table for an AND gate. And that's exactly what happens. In the presence of both inputs, so both signals, our template gets cut and the output is 0. In the presence of only one of the inputs or no inputs, our template remains intact and the signal is present. Now, does it work? Well, obviously, if I'm showing it on the slide during a talk, it does work. It works pretty well, actually. It's pretty sensitive. It's based on fluorescence. It's color. You can design whatever color you want because the fluorescent aptamers have been made in pretty much any color, compatible with standard filter cubes, standard microscope sets, standard plate readers. So in the absence of both inputs, we always have signal. If we add both inputs, we have no signal. We also did a ton of mismatch tests. Shout out to my undergrad Chelsea, who spent basically her entire summer pipetting different mismatches. We have those whole tables to show that this actually is very sequence specific. So it's not sensitive to mismatches, and it's pretty sensitive with the correct input oligos. We have signal or no signal as expected. Now, one logic gate, okay, that's not fun. I mean, that's what can you do with a one logic gate? Can we actually layer those gates? So we developed a system that allows us to layer those gates. So we have a solid state support, a bead or a microfluidic system. Onto that solid state support, we attach our gate. So this is our gate template like on the previous slide. But now this gate template, instead of a reporter oligo, so instead of the fluorescent aptamer, has what we call a signal oligo. So we do our magic. We add 2 inputs or 1 input or no inputs, and we do, oops. So if there's no cut, if we add 1 or 0 inputs, there is no cut. So we do a wash, and then we attach what we call a signal release oligo. So yet another oligo, plenty of oligos in this paper. We add a signal release oligo that then gets cut by another different restriction enzyme, releasing that little guy that we call signal oligo. So that's a signal to the next gate. So if there is no cut happen here, on the previous slide where the output was fluorescence, you could see that if there's no cut, the output is fluorescence. Here, if there's no cut, the output is this little signal oligo. Now, you can imagine what happens if there are 2 inputs. The whole thing gets cut off in the first step, and then when the signal release oligo comes in, there is nothing to release because that signal oligo has already been washed away. So there is 0 to the next gate because we don't have that little oligo. And so we show that it works. We can layer gates. We can layer 2 different kinds of gates, and it's pretty sensitive. It's also very finicky. This is the part that we're still working on to perfect that system. Is in the first example of a single logic gate, the mismatch tolerance is very high. If you have few different gates on the same set of beads or in the same microfluidics chamber, you do get a little bit of crosstalk, and we're trying to figure out, that's kind of one of the things that the real bioinformatics can help us with this time, is to figure out what are the rules for those mismatches. How do you kind of go around how do you go about eliminating mismatch, and also how do you go about figuring out what's the possible kind of a design rules for it. And speaking of design rules, we have a website. Everyone has a website these days. We have a website for the Trumpet project. You can design Trumpet oligos. So if you just want to play with those gates, you can put in your requested gates into the website, and the script spits out the list of oligos you need to order in order to play with your Trumpet systems. It's still a pretty toy system. We have demonstrated all universal logic gates in the trumpet system. We demonstrated the layering, but we're still nowhere near even something like a simple adder. That's the direction that we're going in right now, is trying to make this system more robust, more complex. This was the first proof of principle that you can use that platform to encode logic gate operations, but we're still not to the point where we can say like, yes, I can do fancy computations. I can send it in a rover to Mars and it's going to analyze the environment and whatnot. But you got to start somewhere. Think of it as this is like this very first example of it working, and now we're open to collaborators. People are asking us, what can we encode in the trumpet system? And we're trying to do microfluidic systems too, to kind of be able to spatially separate those reactions to increase the sensitivity. And we're also using it for practical applications. My lab has kind of a rule that we don't do anything practical. We do the foundational work and then we move on to the next shiny toy. But this is something that actually is pretty practical. And so we're using it to develop a diagnostics platform. So using those logic gates, we can pick up short circulating RNAs in the bloodstream. I know I sound like a Theranos founder right now. We can take a drop of your blood and detect if you have cancer. But we're, maybe it's not going to be a drop, but we are developing it into a pretty sensitive diagnostics platform. So this is where we are right now with the trumpet system. Now I want to switch gears for a moment and show you a few different examples of what else can we do with synthetic cells that's not precisely related to running logic gate operations, but is the capacity building. So it allows us to build those synthetic systems to the point where we actually can reach those this big promise of being able to interface with biology, understand biology better. So what is it really good for other than the cool stuff that I was talking about? So tool development, that's one thing that there is a lot of overlap between synthetic cell engineering and developing tools that ended up being useful for the entire bioengineering community. And one of the best examples that I cannot take credit for, but I love using, is the example of the Gibson ligation. All of you who've ever done any cloning have used this cloning technique called Gibson ligation, which is now one of the most commonly used cloning techniques. And it was developed by Dan Gibson when he was working at JCVI on the first synthetic cell, the first genome of the synthetic cell. So the tools that we make to build synthetic cells then end up being really useful for other areas of biology. Another example is nanopore sequencing. Probably all of you have heard, all of the nerds here have heard of the DNA sequencer that was sent up to the ISS. That was a nanopore-based DNA sequencer. And that was developed, but the technology was developed based on an alpha-chemolysin channel, which is the most common channel that we use in synthetic cell engineering. And the guy that developed the nanopore sequencing, now it's not alpha-chemolysin based anymore, but the original first prototypes were. And the person that developed it originally was a synthetic cell engineer that was studying alpha-chemolysin for the purpose of making pores in a synthetic cell. So the tools we're making actually end up being useful for everyone. Another thing is understanding natural biology. The Feynman said, what I cannot build, I cannot understand. And the great lesson from that is we still don't understand biology because we cannot build it. And we want to understand biology because we are biology. I would like to be able to understand how both healthy and disease processes work in my own cells so I can make better drugs, so I can live to be 100 and whatnot. And that's part of what motivates us, is we want to reconstitute all of those biological processes in a synthetic cell system so we can truly understand it. Metabolic engineering is something that really makes money these days. We make metabolic engineering circuits to make molecules, to make small molecules. That's where the biocomputing really comes in, because all of those metabolic engineering circuits need to be controlled. And control is something that we're really lacking right now. So that's where most of our bioinformatics collaborations come in. Diagnostics and therapy, another moneymaker, but also useful. It shows that our field actually produces something that's useful. Biocomputing, I just showed you an example. Space exploration, origin of life. These are the cool out there applications that really drive the imagination. For space exploration, all of the examples of reliable biocomputing to make autonomous machines, but also bioproduction during long-term space missions. Origin of life, how did life start? We can figure that out by building it. Last but not least, safety security. I'll talk about that in a few minutes again, because we want to make sure that everything we're making actually remains contained and remains safe. So I'm going to show you very briefly a couple examples of work out of my lab that shows the utility of each of those kind of applications. So one thing in the tool development area is that we asked, can synthetic cells produce something that then can be exported outside? And if it's exported outside, can it go into another cell? And I'm sure many of you recognize that this genetic circuit that's shown here is a simple AND gate. So this is the intersection with biocomputing, is we can make those simple biological circuits in different populations of synthetic cells, between populations of synthetic cells, using proteins that are made in one population, exported outside, and then imported into another one. And that gives us a lot of control, not just over those genetic circuits in synthetic cells, but also in interfacing with natural cells. So you can imagine doing biocomputing in a kind of a distributed way where you spread your circuits across a few different populations and they exchange the signals by protein. It's going to be slow, it's going to be inefficient, but it's going to be made out of biological parts, which means it's renewable. And if you can actually program it to be kind of a not very leaky, very reliable, then you can have this kind of a slow going, slow boiling computational system. Another thing that we wanted to do is look at antibiotic resistance. So the prediction is that by 2050, the most common cause of death in developed countries will be antibiotic resistance diseases, because we're really bad at making new antibiotics. So that's something to look forward to. And we're really trying to understand those processes. And again, from the computational angle, antibiotic resistance is really a kind of a logic gate type operation. Here comes a signal that's a small molecule antibiotic. And what a cell does with it is your logic operation, whether that antibiotic causes death, sloth of growth, or it does nothing. And so to understand it, we really need to build kind of a molecular model for it. And that's what we did, is we built a molecular model of antibiotic resistance. So we have two populations of synthetic cells. One of them is resistant to antibiotics, the other one isn't. We add antibiotics to it, and the resistant one expresses proteins. The not resistant one gets dead, or even deader, they're already dead. Now we can also add membrane channels to let the antibiotic in, some pretty microscopy pictures to show you that those things actually exist. And the channels, the membrane channels in the membrane of the synthetic cells allow us to impart resistance or a susceptibility to antibiotic to different populations of synthetic cells by exchange of those channels. So this is kind of like programming whether the circuit inside the cell is taking up antibiotic or not. And we can also do some magic where we can mate two different populations of synthetic cells together. We call it mating. It's not really mating. It's programmable fusion, but we call it mating because it publishes better if you call it mating. The biologists will like it. And what we can do is by this cell fusion, aka mating, we can impart resistance or susceptibility to antibiotics. So this is kind of a system that allows us to model different behaviors in presence of small molecules that affect the circuits inside a cell. We did antibiotics because that was kind of our proof of principle, but you can also imagine any other small molecules that can control circuits inside cells. Okay, so here's an example of metabolic engineering. And again, it's going to be a circuit. Synthetic biologists love circuits to pretend that we're biohackers. That's the word everyone likes to use. So we love to call those things circuits. So what we have is a cell-free protein expression system that's been immobilized on a solid support, so literally pieces of paper, and lyophilized. And if you lyophilize it, you can store it. We have data that shows that this can be stable at room temperature for up to 3 years. And so you can store it at room temperature lyophilized, and then you can combine your pieces of paper. So solid state support is the fancy name for it. It really is a paper. So we can combine pieces of paper together and reconstitute a genetic circuit that produces fluorescence. So it's the same principle that I showed you in case of the trumpet. We have those genetic systems that produce a fluorescence. It produces fluorescence only if you reconstitute that circuit and add water. And that's where the combinatorial part of metabolic engineering comes in, is we can build circuits that produce different small molecules based on what those single pieces of paper are. So imagine it's basically a paper sandwich that we put together, and when we put it together, it can produce, this is an example of a simple metabolic engineering circuit. It's a bioacetic acid circuit. Those of you who've ever talked to metabolic engineer, you will know this pathway because that's a favorite proof of principle pathway of all metabolic engineers. It makes bioacetic acid, which is pretty colorful. It analyzes well on HPLC. So we demonstrated that you can use those systems to basically make this combinatorial bioengineering pathway. And so the idea is that you can store elements of your pathway at room temperature as pieces of paper, and then you can reconstitute different pathways depending on the order in which you put those different pieces of paper together. And that's useful for things like long-term storage, low resource environments, but also things like space exploration. Because if you send Elon Musk to Mars, eventually he will need medicine, and that medicine cannot be delivered. It has to be produced on the site. So having this kind of agile room temperature biomanufacturing platform would be really useful. Now, couple examples of things that synthetic cells are good for that are not work out of my lab, but I think is really worth showing because it shows you that this technology can actually be useful. This is work out of Avi Schroeder's lab at Technion. They demonstrated that with synthetic cells, you can shrink tumors in live mice. So this is where this technology is right now. You can take synthetic cells, inject them into a mouse with a breast cancer tumor model, and that tumor will shrink. It also is possible to use synthetic cells to grow vasculature in an organoid. It's possible to use lyophilized cell-free platforms. So this is what I showed you a few slides ago with that lyophilized paper-based system to produce vaccines. And those vaccines actually do produce immune response in mammals. That's work out of Jim Collins' lab. And last but not least, Fritz Simmel demonstrated that it's possible to have a synthetic cell system that produces replication-competent viruses, including bacteriophages. So it's not just model proof of principle fun technology, it also has some useful applications. Okay, now forget useful, let's go back to fun proof of principle stuff. We talk about using this for long-term space exploration. So the question is, does it actually survive under space-like conditions? So we designed two experiments, a protein expression system and a functional RNA aptamer system. And we loaded it up on a CubeSat and we launched it. And I'm showing that video because I had to be up at 3 a.m. to watch that launch. Also, it's my own sample going into space. I mean, how cool is that? To me, that was really cool. And so it did, oh, the video's not playing. Let's see. Imagine a rocket launching right now. It's going up. It's nice flaming, but it doesn't explode. My sample goes up, and it was a sample return mission, so we got it back. The video was great, beautiful, and whatnot. Always check if your videos are playing when you upload it on a foreign laptop. Clearly lesson learned. Okay, so a rocket went up, rocket came down. We covered the sample. Yes, it works under space-like conditions. We can send lyophilized system up, reconstitute it with water, and it will work. Then a couple years later, we send it up to the ISS in collaboration with BioBits, which is a great private foundation that develops cell-free systems for educational purposes. So we sent some of those lyophilized systems up, and we actually had a human astronaut do the pipetting to set up those reactions. So it's possible to assemble those circuits using actual hands, not just the robotic microfluidic system like here, and it still works. So it's pretty robust. It survived. Now, what's another kind of a fun proof of principle, useless but exciting application of it? Mars is not really a good environment for life. When you think about terraforming Mars, you've watched all those movies where Mars becomes green. That will be extremely difficult. Not for all the obvious reasons that you can think of. Like you watch this movie where a guy grows potatoes on Mars. That's also impossible, not for the obvious reasons you can think of. It's impossible because Martian regolith is essentially a brine of perchloride. Perchloride, the same thing that's in your household bleach. So Martian regolith has a very high concentration of perchloride. And most environments on Mars that we sampled so far are chock full of perchloride. Life is not very compatible with perchloride. So we set up to test if it's possible to do lifelike reactions in perchloride conditions that are compatible with the Martian regolith. Obviously, we didn't have a sample of Martian regolith. We had to simulate it. but we, I think, simulated it close enough based on spectrophotometrical data. And so we demonstrated that you can do some of those synthetic RNA and functional RNA reactions in under conditions that would be compatible with wet Mars. So you still need water, but it's compatible with the Martian perchlorate. So it's possible to imagine origin of life processes that happen on Mars back when Mars had water and it was much warmer. There's probably no extant life on Mars right now. Any life that's there is probably from the junk we crashed on there, but there could have been one at some point, and it's also a good lesson on how you model extreme environments, how you model extraterrestrial environments for looking for life elsewhere. So when you go to Enceladus and Europa to look for life there, then we will know how to model those reactions. Last but not least, biosafety and biosecurity. If you're building biological systems literally from scratch, you want to do it in a way that is contained. And so one of the examples that we very recently investigated of making a synthetic, one particular type of a synthetic cell that could potentially not be contained is this so-called mirror life. So we've, my lab included, we really wanted to make a mirror cell. We wanted to make a cell that contains all of the same biomolecules as a normal cell, but they're of the opposite stereochemistry. And then working with immunologists and ecologists, we realized that we actually should not be doing it because that thing would be uncontained and could spread in the environment uncontrollably. And so we came together as a scientific community and we said, please don't do it. We're calling for a global moratorium on the mirror life research now. And that's a good example of how this field can self-organize. We're not just blindly running with this research saying, this is cool, I want to do it, so therefore I'll do it. We're actually thinking about what we're doing. We're analyzing safety and security implications of what we're doing, and we're trying to make sure that what we're doing is actually done responsibly. And the reason why this is so important is because pathogens know no borders. Biosafety and biosecurity is a global problem. Even if the regulations in one country are excellent and all the scientists obey those regulations, if someone across the border lives in an environment that's less regulated, if they have an accident or they do something maliciously, this will get out and will not be contained. That's why both biosafety and biosecurity in any engineering biology project have to be treated as a global problem, because pathogens don't know borders, and also what doesn't kill you will mutate and try again. So accidents, near misses in the lab are actually a big problem, because if something gets out of the lab, even if it's not pathogenic at the moment of its release, it can become pathogenic when it spends enough time in the environment. So we all have to think about it. And by all, I mean there is a great international community of people considering all the aspects of engineering synthetic life. We call ourselves Build a Cell. This is based on the Build a Bear initiative. We had a workshop. We have workshops twice a year. One time we had a workshop in Austin, and for each workshop, we put out those big signs in front of the building that says Build a Cell workshop here. And one of my co-founders of this initiative, he was coming up in the Uber, and the Uber driver said, "Oh, Build a Cell, yes, more jails, yes, go for it." So that's Texas for you, but it was a great workshop. The Build a Cell community is a international, open, friendly community that builds biological cells, that I have to emphasize that, builds lifelike organisms. What we do is we do not define life. So we do not tell you what you can or cannot build. We do not limit ourselves. What we do is we share results, we share unpublished results. A lot of the time you probably wasted a lot of time trying to reproduce someone else's experiments, or you have an idea and you try it for 6 months and it fails. And then you go to a meeting and you talk to a colleague and he says, "Oh, we actually tried that 2 years ago, it didn't work." Well, I would love to know that before I wasted time trying it. There is no way in any scientific community right now to effectively share unpublished data. That's what we're trying to do through Build a Cell. And the main mechanism that we're using is we actually meet in person as a community. We meet twice a year for workshops. It's been an amazing run. I just came back from workshop 14. We have always amazing attendance. People still believe in this collaboration, in doing it as a community. We look at science policy. This has been iffy in the last few months in this country, but we try to influence science policy in the ways that actually support research while safeguarding it at the same time. And shout out to NSF for funding us, buying us coffee and travel and plane tickets, and supporting that amazing community. And last but not least, I want to thank to everyone in my lab. When I started my lab almost 6 years ago now, I said I want to make cells from scratch. And people actually believed me. They gave me money to do it. And most importantly, amazing people joined my lab. So I want to say thanks to everyone who funded our work and thanks to everyone who worked with me in that period and who contributed to all the data that I was able to show. And thanks to all of you for listening, and I will take questions.
Thank you. We have time for a few questions.
Thank you.
Thank you. That was so much fun.
Thanks so much.
So you started out by motivating this by pointing out that you would like to have cells that do things, but that also can repair themselves under, and to me, that sounds like evolution.
Mm-hmm.
But you've also pointed out that, if you let it just sort of do whatever it needs to do, that it's going to evolve away from your original goal. So do you have any thoughts about that conundrum or that balance there?
We're hoping that once we're able to engineer evolution, it will be a controllable evolution. So we'll be able to control mutation rates. We'll see how it goes. We have a couple projects, and other people in the field have a couple projects where we're able to control mutation rates pretty tightly, but it's not to the point where we can actually truly call it evolution yet. I'm thinking, when I think repair, I think more of a replacing existing components. So it wouldn't be agile enough to react to the environmental changes. It would be more like, this is my architecture and I will maintain it no matter what. And if I get bombarded by radiation or salted out or whatever, I'll persist and then remake it. Not that if...
You might have to adjust to different environments, though.
Yeah, so the adjusting to different environments might be the engineering part. It's possible that we will never be able to have a synthetic cell that will just truly evolve, adjust to different environments on its own, which wouldn't be the worst, because that means it would still be contained.
Mm-hmm. Thank you.
Thank you so much.
Thanks. I'd like to echo what Danny said. This was incredibly fun and very, very informative and very huge in scope. I have a question about something that always confused me when Mirror Life came out and everybody says, no, don't do it. My thought was, mirror amino acids and mirror sugars, we don't have a lot of those in real life, and they wouldn't even interact with not real life, with existing life on Earth, and they wouldn't even interact with us. So
Yes.
Why do people think that might be a problem, that might be pathogenic? Although there's the unknown factor, of course. Yeah. Which brings me to my second question, which is, what are your thoughts about, you probably know the shadow biosphere hypothesis, where, we have, you might say we have our current life, and then there will be this very cryptic microbial life that doesn't interact with us because, water and oil, they won't interact, like mirror isomers. So these questions are kind of related. Thanks.
So to answer your first question, the concerns about mirror life are not because it would be an active pathogen. The concern is because it would not interact with the rest of biology. So it would not have predators. It would not be attacked by phages. Nobody would eat it. And also your immune system would not recognize it. So it would be an organism that can use chiral carbon sources in the environment and just grow. Even if it's a very wimpy organism, it grows really slowly, but it grows basically undeterred. It can grow and it will not be susceptible to any of those regulatory mechanisms. No viruses, no predators. If you got a mirror life infection, your immune system wouldn't know what to do with it because it will be stealth to the immune system. So it would just keep growing. And yes, it will be susceptible to the mirror antibiotics, but you cannot throw antibiotics all over the biosphere. So it's more of a ecological concerns, even though human health concerns are there too. And to answer your shadow biosphere question, I would love to find one. I hang out with a lot of physicists that pretend to be biologists, and they say it's impossible to imagine a biosphere, a single planet supporting more than one biosphere. And they have complicated arguments. The word Shannon entropy shows up a lot. No one knows what that really is. The idea is that one planet means one biosphere. That seems to be the paradigm among the people that look into it. I don't believe that. I think it's possible to imagine a shadow biosphere, a physically separated shadow biosphere. And I'm still optimistic that we might be able to find one, although the number of environments is dwindling.
They do have an N of 1 for their hypothesis.
Yes,
Yeah.
that's true. And all of biology has N of 1
Yeah.
so far. So that's the problem with all our hypotheses.
Thank you very much.
Thank you.
So this is an online question. Washing appears to be needed for the signal oligo version of the trumpet system. Is there a more independent approach? And then also regarding self-replication, how possible is it with synthetic cell systems? Are there any successful attempts?
The first question was, can we do without washing in the layered trumpet gates? We tried, but we don't have much luck. We tried to do like a 2-bead system where the second oligo goes into the second bead. We haven't had much luck with it. We're right now washing it in a microfluidic system. So the washing is kind of a part of the process, and but yeah, we're still stuck with the wash steps. The second question on the self-replicating synthetic cells, we're very close. There is a Petra Spille's lab at Max Planck who demonstrated that cytoskeleton, artificial cytoskeleton, can create almost like a budding yeast type system. And then we also have a system up and running in my lab that produces a complete cell cycle and a genetically encoded, very low yield replication. I wasn't showing the data here because you guys are not biologists, so you're not supposed to be interested. But yeah, that's kind of where the field is right now, is we're very close to making a self-replicating synthetic cell, but we're still far from making a robust synthetic cell. You know, you can make something self-replicate under perfect conditions. That's not that difficult. The difficulty is to make it actually robust enough that it can replicate in the environment. That's I think where the real threshold is, and we're not there yet.
It was really fascinating. I'm
Thank you.
sorry to ask kind of a prosaic question,
Please do.
but I was intrigued by the sort of like paper, the lyophilized like paper system, and it reminded me of like, you know, really like old analytical methods like paper chromatography and stuff like that. So could you kind of like combine that, like breathe new life into those methods, especially for like you mentioned low resourced environments?
Absolutely.
Yeah.
I'm more on that particular sandwich project. We work with a collaborator, Lynn Rothschild at Ames. She works on developing. She actually is a card-carrying space biologist. She works with flight surgeons on developing technologies for flight, especially for flight medicine. And she calls it paper-based microfluidics, where they do exactly what you described. It's like the old-fashioned chromatography. So we make a small molecule, and then her paper-based microfluidics separates it. She has like papers with almost like immobilized purification beads in a paper form. So if the protein of interest is his tagged or flag tagged, it can be purified in this very kind of a low resource environment.
That's really cool. Thank you.
We've also, another example is we've demonstrated that some of those transcription reactions can happen on a piece of paper, and they don't happen at room temperature very well, but they do happen very well when you heat them up on the inside of a human armpit. It works. My grad student made the great sacrifice of taping it to the inside of his hand and walking around with it for a few hours. And it does work. So it's possible to do reactions like that.
Listen, like low-cost analytical chemistry, I'll stick something in my armpit.
Yes, we don't do mouth pipetting. That's where I draw the line.
Okay. Thank you.
Thank you.